This is the video that is 100% mine. It's not in any biology book, but like I said, there are problems with evolution. Just because some people take it like it's written on a stone, it doesn't make it so. With any other theory, if you have problems, you adapt the theory. But something about evolution is like, no, this is what I've always heard, and I'm not changing my mind, and you cannot make me change my mind. But the fact is, there is more and more scientists every day that believe the theory of evolution should be at least modified. Okay? If you don't believe how many, I suggest you go to a website. It's called Descent from Darwin, and look at the list of people who sign it. And if you look it up, you'll find my name in there. Okay? I'm proud of that. But it's only scientifically honest to show you, like say, both sides of the controversy. This, we saw it in the video about evolution as evidence for evolution, but we can dispute all of them. There's problems with all of them. And instead of addressing the problems, a lot of people choose to just close their eyes, close their ears, move away. What's wrong with you? How do you dare? All I'm doing is asking you, can you explain this to me, please? No, don't do that. We talk about fossils, okay? But there's a lot of missing fossils, usually transition organisms. Let's say that you have an organism A that starts evolving, it becomes B, C, D, E. What a coincidence. We have fossils for A and fossils for E, and they're very, very different. But you don't have any fossils left for B, C, and D. And people will tell you, hey, these fossils are very delicate when they're forming. And not all of the circumstances always work out where an organism can fossilize. They need very specific conditions. So we don't have fossils of everything. I agree. What a coincidence that you have the first one and the last one, but you don't have the ones. And not only happens with this one, but it happens over and over and over. You always have what they call is the original one, what they call the final, and they say, but we're missing everything in between. And it, like I say, happens over and over and over. It's too much of a coincidence, isn't it? And like we said, when we talk about paleontology, there you have examples of fossils from the Cambric explosion. Fossils are evidence of the Cambric explosion. You go from having one or two organisms to having a bunch, but they don't explain it. Okay? But let me give you better examples. Polystrate fossils. They're usually tree trunks, and they're more common than you imagine. Okay? But they're embedded in a fossilized rock. And they said the rock is maybe, I'm trying to remember the particular example I got from the internet. I think this one was, said the rock is 50 million years old. Slowly, material was being deposited and eventually created a rock. So you're telling me that this tree trunk stood exposed to the weather for 50 million years? Come on, when a tree dies, it's a matter of time before the tree starts growing. The wind blows and starts breaking it. Eventually the tree falls. And you're telling me that this tree trunk stood there, perfectly intact, for 50 million years? Okay. I have to laugh, okay? I think the fact that the tree trunk is still standing is evidence that this rock was actually laid down in what some people call the geological instant. It's not an instant the way we understand it, but in geology we talk about millions of years. So something that takes weeks, maybe months, would be considered an instant in geology. Okay? But obviously that tree trunk could not be exposed for millions of years. End of the story. And there's no explanation. And like I said, there's plenty of examples of this. Another of my favorites, the coelacanth. We had the fossil, and people said it was extinct 60 million years ago. But if you look at the flippers, they have a bone that looks very much like the leg of an animal, and then it has the rest of the fin. So they say this is a transitional species between the fish and the four-legged animals, the tetrapods. We finally got a fossil of a transitional form. Hey, doesn't that prove evolution? People think that, and it still appears in biology books, but in 1938, a South African museum curator found a coelacanth. Ever since then, they have found many examples of living ones in Asia. And you know what? After supposedly 60 million years, it still looks like a fossil. It never grew legs. It never got out of the water. You know what? It was not a transitional form. Because in 60 million years, if you believe what they said, it never changed. This is more modern. A number of dinosaur fossils have been found with soft tissue. The example I'm giving you here, the phomelanine, it was a fossil that had been dated between 75 and 100 million years old, under the best conditions in the lab, in an atmosphere of nitrogen, and in a deep freezer, blah, blah, blah. If everything is perfect, you can keep melanin a few thousand years, not 100 million years. And it gets better, okay? If you look at the recent studies, they have found red blood cells in dinosaur tissue. And you know what? The cells have lost their shape, but they still have hemoglobin. And that hemoglobin is still good enough. It still has the heme group that attaches to oxygen. Why didn't the hemoglobin decompose? Hemoglobin doesn't last very long. Think in your body, and you hit your arm accidentally, and in hardly no time, you get a bruise. That bruise is the hemoglobin is starting to decompose. But you're going to have hemoglobin that has preserved for millions of years, and they keep finding more and more examples, okay? But the soft tissue puts a big question mark on the millions of years of age. I don't want to confuse you with radiometric dating, but remember, I'm both a biologist and a chemist, and I'd like to spend a lot of time talking about that. How do they do carbon-14 dating? Well, basically, all organisms have carbon-14 that decomposes into carbon-12. Okay? It decomposes or radioactively breaks into decays into carbon-12. But the amount of carbon-14 in an organism is constant. I'm sorry. Hey, when the pen does that, and I'll never learn. Okay, sorry. Okay, but the amount of carbon-14 remains constant because you're taking in carbon-14 from the air at the same rate that it breaks down to carbon-12. So as long as an organism is alive, there's a certain ratio of carbon-14 to carbon-12. But once the organism dies, it stops taking in carbon-14, so the ratio changes, and that allows you to date how old the organism is. But they make a big assumption. They say, you see all of these old trees, something like that? There was always the same amount of carbon-14 in the atmosphere that there is today. We're assuming that they all began with the same ratio of carbon-14 to carbon-12, and that ratio depends on whatever was in the atmosphere. But there was a time when there had to be more carbon dioxide in the air, which means more carbon-14, because remember, in the Carboniferous, you had those giant trees. They were ferns as tall as trees. Plants do photosynthesis. They need carbon dioxide to grow. So I know there was nobody alive during the Carboniferous, but don't you think there's reason to expect that there was more carbon dioxide in the air than there is today? Which means all of our calculations are based on a wrong assumption. People look at a rock and say, okay, when that rock was formed, there was no lead in it. All of the lead that you found today comes from uranium that had decayed into lead. So looking at how much uranium there's in the rock, how much lead, we can tell you how old the rock is. And my question is, How do you know the original rock didn't have lead? Who told you that? Were you there to test it? Was anybody there to test it? You're telling me it's 50 million years old? Who tested it? You're based on assumptions. I'm not saying whether right or wrong. I'm just saying you're just assuming that. You don't know. So quit talking like that's a fact. It's just an assumption. In fact, there's a recent study of radioactive decay of uranium in New Mexico, and they say that about 6,000 years ago, the rate of decay was about 250,000 times faster than it is today. I'm going to be honest. I tried to read the article. It's way, way above my head. All I can do is take their word. I cannot tell you that, in my opinion, this is right because, like I said, extremely complicated. But there's some suggestions that maybe radioisotopes don't decay at a constant rate. We talk about argon-40 as being one of the ways to date rocks. Well, they found out that whenever there's lava, there's a very large amount of argon-40 in lava, and that contaminates the samples. It's adding extra argon-40, which makes it look like the rocks are older than they really were. Because the sample was not isolated. Argon-40 was being added continuously in there. And of course, you have to agree, Einstein said the universe is expanded, time is relative, so the time that we're measuring today is that the same thing that time that took place however many thousands of years ago? At least you can agree there's a question in there. Okay? But radiometric dating, this is just a very quick overview. It has a number of flaws. And many times, I told you before, people use the wrong technique. People who claim to use carbon-14 to date a rock. Sorry, but rocks don't have carbon-14. So I don't know if you made a mistake typing, or use the wrong technique, or you're trying to deceive me. I don't know. But what you're telling me doesn't make any sense. We talk about homologous structures, having that they have similarities, and they say that's because there was a common ancestor, and then the different species that came out from that ancestor adapted to different environments. Could it be that they look similar because they have a common function? In this example, it could be movement. And you say, well, we don't move with your arms. Okay, I agree, but I didn't have a drawing of a human leg. But the human leg is going to have one long bone, then it has two little bones, then you have your ankle, and then you have long bones doing the foot and the toes. It's the same pattern, okay? The pattern on the leg, the bone pattern, is the same that you find in the arm, the same you find in the cat, in the bat, in the dolphin. And they all have a function, movement. If you go a little bit more in detail, why do you have two bones in the forearm, on the lower part of your leg, something like that? Actually, because of having two bones and because of the heads of those two bones, I'm not going to get into too much detail, but that allows you to rotate. Put your hand flat on the table. You can rotate it, put the palm of your hand looking upwards, but your elbow didn't move. You have a rotation in your forearm that is possible only because of those two bones and the shape of the heads of those two bones. That's a function that's common. You're going to find in all of these examples. So maybe, I'm not saying that's the reason. I'm just saying maybe they're similar because they have the same function. And if it works, don't fix it. We talk about homologous structures. Okay? And if you look at the embryos, actually the embryos divide into segments. I'm not going to go into too much detail. You'd assume that a particular structure, like the legs, are going to appear in the same segment, being controlled by the same genes. I know this diagram is not very easy to see. I couldn't find a better one that showed this, but you can see that the legs in different organisms appear from many different parts, many different segments, and every segment is controlled by a different gene. Homologous structures, many times have been shown not to be produced by homologous genes. So if there was a common ancestor, how do you explain that? Wouldn't the same gene control the arms and the legs? But they don't. So it's very difficult to explain common ancestor in this case. We talk about vestigial structures like the hips and the well, and we talk about the appendix. Appendix has always been the example of a vestigial structure. It doesn't have any use other than make you sick, right? But now we know that it's part of the lymphatic system. It contains lymphoid tissue. That means it has white blood cells ready to attack an infection. Now, there's very few bacteria that will survive the whole digestive system and get in there, but some do. And you have a standby army of white blood cells ready if any bacteria gets into the large intestine to attack. The fact is, there was a time when at the size of the thing they removed your appendix. A recent study showed that people that have had the appendix removed were 2.5 times as likely to develop intestinal infections as the people who still had the appendix. Why? Because the appendix is part of your immune system. The same thing with tonsils. There's a time all the tonsils do is cause trouble. They cause strep throat. When I was growing up, it was almost automatic. You get a strep throat, we'll remove your tonsils. That's fine. You know what happens? That's part of the lymphatic system. They protect the infection from getting from your throat, where it's easy to treat, down into maybe your lungs, into the bronchial tree, maybe the lungs. So people who had the tonsils removed didn't get a strep throat. They got bronchitis, they got pneumonia, they got far more serious diseases. And now it takes a relatively large number of recurrent tonsil infections before a doctor will remove it, because it's not a vestigial organ, it's not a useless organ. So we use that as a proof of evolution, but there's no such a thing as a vestigial organ. That's what I'm trying to say. As far as embryology, we talk about Haeckel and his drawings, and that was supposed to be proof of common descent. While he was still alive, he was charged with doctoring the images. They said he invented features that were not observed in nature. People actually went to his lab because he claimed to have all of the embryos. And the embryos and the drawings were similar, but not identical. He added things. He said, oh, I didn't really add it. I just emphasized this structure so you could see it better. Fact is, his drawings were not accurate. And while he was still alive, it was proven that it was a fake. I could go even further and say, he studied at this particular age, but if you look earlier in here, those embryos are very, very different. So embryos of different species, very, very young, are very different. Then they seem to come closer, and then they start separating again. But by cherry picking this group and forgetting about the older ones, the younger ones already earlier, they were able to prove something that wasn't there. What is very aggravating is that it's been over 200 years that have been proven that Haeckel had doctored those images, but you still find them in textbooks, you find them in museums, still proof of evolution. It's just, it leaves me speechless. I mean, if you prove something to be a fake, how can you still show it in textbooks and say, this is a fact? I told you, people are not reasonable when it comes to evolution. That's the only explanation. Then we talk about, okay, let's look at macromolecules. I mean, this is the 21st century. Let's forget about drawings and all of that. Well, we said that humans and chimps have been said to share 98.8% of the DNA, which meant that they were very closely related because there was a common ancestor between humans and chimps. Like I said, they didn't look at the entire genome. They only look at the part of the genome that codes for proteins. A 2025 study, that's not all at all, showed that the similarity is more like 85%. When you consider how many genes code for the same type of protein, regardless of what the organism is like, any organism that has blood needs hemoglobin. Well, you're going to have the gene for hemoglobin. You're going to need the genes that keep a cell going, cellular respiration, blah, blah, blah. 85% similarity is not amazing at all. It certainly is not proof there was a common ancestor between humans and chimps. Some people say, well, forget about it. We're going to look at the proteins. And the fact is, if you look at protein sequences, they're more similar than DNA between species. But you can still find exceptions. Look at this particular protein, cytochrome C, that's part of the electron transport chain. And they compare the rabbit, the guanaco, that's similar to the llama, and the horse. The guanaco and the horse are hoof ungulates. They're closer anatomically, phylogenetically, whatever. But if you look at the rabbit, it's completely separate, different thing, doesn't have hooves, something like that. But look at the differences and say between the rabbit and the guanaco, there was 2 amino acids difference. Between the horse and the guanaco, there was 6. So that doesn't make any sense. You're telling me that the animals that are closer together have more differences? Maybe, just maybe, protein sequence does not tell you how close some organisms are. Some scientists have said that actually the similarity between proteins is an example of efficient design. Compare that to a computer programmer. Okay? If you need to write new code, you don't start from scratch. You can sometimes take the old code, change it a little bit, tweak it here and there, and then you can do a different job. Is it possible that these proteins, they have been tweaked a little bit to explain the difference between different animals, but they're similar because it's an efficient design? Now, I understand if you're talking about design, we're going to say, well, wherever there's design, there has to be a designer. But what I'm trying to say is that protein sequences and DNA sequences do not always match with the phylogenetic relationship between organisms. They do not prove common ancestry or evolution. So what would it do for me to tell you there's problem with evolution if I didn't tell you what many scientists believe would be an alternative explanation? Okay? And the most common one is intelligent design. Now, I understand some courts have said you're not supposed to teach intelligent design in a public institution because it's too close to quote-unquote religion. I don't care what the courts say, okay? This is an explanation offered by scientists that makes some sense. I'm not saying it explains everything, and it's only honest to see what they have to say. What these people are saying is that certain features in the universe are too complicated to be explained by random processes like natural selection. They need an intelligent cause. The cell, look how complicated the cell is. Remember that. What are the chances that all of the things were developed at the same time and they came together in the same cell? Because if you don't have all of the components of the cell, the cell won't live. That's what they're saying, that the cell is actually a proof of intelligent design. It doesn't mean there's no evolution. That's not what they're trying to say. They're just saying that what you consider evidence of common descent does not necessarily mean natural selection. There's something more. There's an intelligent cause behind all of this. When we talk about the proteins, we said, we compare to a code in a computer, and we said there's a coder that reduces an old code and tweaks it because it works, just adapted to different parameters. Intelligent design basically saying the same thing. There's an intelligent cause behind what we are observing. And this is a very simple example, but I think it shows you, because they base everything in irreducible complexity. Basically, you have a system that's composed of several interacting parts. They all contribute to the basic function, and if you remove one of them, the system stops working. And the typical example is a mousetrap. Imagine you don't have the holding bar. If you don't have the holding bar, you have no way of holding the spring, so the trap will never be set up. How would it catch a mouse? It couldn't, unless you tell me the mouse just happened to come by, trip over the trap, fell off a table, and broke his neck. No, the trap cannot kill a mouse. The same thing, if you don't have the part that moves the hammer, it's not going to catch anything. If you don't have a catch that holds the trap set and puts the bait, do you understand? If you remove any part of the mouse trap, it doesn't work. The same thing with a cell. If you had a cell without mitochondria, it couldn't do cellular respiration. So how did the cell live? You know, those people who tell you the ancestral cells did not have mitochondria, but one day they swallow a bacteria that could do cellular respiration, and the bacteria was so happy inside the cell that decided to stay, and it became the mitochondria. Okay, so how did the ancestral cell live without cellular respiration? How did it get energy? Crickets. There's no answer. Okay? I'm going to show you a couple of examples of irreducible complexity. The bacterial flagellum, not just the flagellum, but the attachment. The bacterial flagellum moves like a propeller in a boat. It takes, depending on the author, between 30 and 50 proteins that function just like a model. I mean, this is an electron micrograph, it's a drawing in here, but basically you have all these parts that you can slowly compare. It works just like a roll and all of that. And in a model, if you're missing a part or one part breaks, it stops working. Same thing in the bacterial flagellum. If you was missing any of these proteins, it would not work. So you couldn't have evolved one piece at a time because you need all of the parts for the flagellum to work. If the flagellum doesn't work, the bacteria cannot move, it cannot get food, it dies instead of reproducing. This is an example of irreducible complexity. Either you have all of it, or you don't have a working flagellum. Same thing, the blood clotting cascade. Obviously, I don't want. What I want you to see is that you have a bunch of proteins, and every time one gets activated, it serves to activate another one. That's what's called a cascade. So you have one protein activating another one that activates another one that activates another one. The final thing is a protein that's soluble and it's in the blood that becomes a polymer that starts forming a clot. You need every single factor in the clotting cascade. If you're missing one, like people with hemophilia, then you don't have chlorine. That organism will not live, will not reproduce. I like the way, this sounds silly, but this is Dr. Behe, the person who published about irreducible complexity, and he said, what good is half a scab? Of course you don't get half a scab. You don't get a scab until you get to the end. He was just being sarcastic in there. Okay? But once again, you're missing anything in here. The clotting cascade cannot get to the end, so you just bleed to death. Either you have the whole clotting cascade, or you don't have chlorine. End of the story. That's why hemophilia is such a serious problem. One thing that people who have studied mutations, especially good mutations, is that even beneficial mutations are caused by loss of genetic information. It's not a consequence, it's a cause. Okay? And I'm talking about good mutations. But you always lose genetic information. Evolution says you're creating new information. When you adapt, you create more information. But people who study and they get mutations and they actually literally sequence the DNA say, sorry, you never add extra information, you only lose information. Remember we talked about polar bears? Polar bears eat fat. They don't have high cholesterol because of some bad mutations in this gene. That gene does not work, but that causes the polar bear to find an alternative way to digest the fats that prevents it from getting high cholesterol. But it was a damaging mutation. There was loss of genetic information. This, when I read Lenski's book, it was published in 2000 and the study was even older, but at the time that the book was published, he had grown 65,000 generations of E. coli. Now, E. coli reproduces very quickly, so he was saying that those 65,000 generations would be the equivalent to 1 million years in humans because of the difference in lifespan. Okay? All of the cells that have mutations have massive deletions. Some of them were beneficial, like all of a sudden there was a particular E. coli that could use citrate as an energy source instead of glucose. That's fine. But when they look at the genes, it wasn't that all of a sudden it developed a new gene. No, it was that an old gene that was present in there was deleted, or at least part of the gene was deleted, and it just happened to be beneficial to the bacteria. But every single mutation that has been studied that leads to an adaptation always involves loss of genetic information, and that goes 100% against evolution. Like I said, not all scientists agree with this. It's up to you what you want to believe, but you cannot know what you believe into unless you have all of the information. So if you want to discuss more about this, if you want to ask questions, remember the video description has a link to my Wise and Turing profile. Make an appointment. It's one of my favorite topics to discuss.